Let's talk neurons. Hooray! So beginning this section, talking about neurons and action potentials and neurotransmission. And in this and the next video, we're going to talk about the neurons and the structure and function of the parts of the neuron. So let's just dive right in. So when we're talking about the cells of the nervous system, there are neurons and neuroglia. In these first two videos, we'll talk about the neurons. In the third one, we'll talk about the neuroglia, which we used to consider just to be support cells, but it turns out they're very, very important. But the neurons are one of the main cells. Well, actually, there's way, 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 way more neuroglia than neurons, but it's an important cell in the nervous system. They carry information. And just some terms, you may know these already, but an afferent neuron is also called a sensory neuron, and that carries information in to the brain and spinal cord. Efferent or motor neurons carry information from the brain and spinal cord to the muscles, the glands, they carry it outward. And then interneurons are in the central nervous system and they connect afferent and efferent neurons. All right, so let's look at neurons and talk about some of the things. So first of all, there's a lot of them. Just in the brain, I think there's about 80 billion neurons, and there's 100 million just in the gut. So we have a whole lot of these. And as many as we have 10 to 50 times as many neuroglia, which we'll talk about. But so we have a whole bunch of neurons, and each neuron connects with other neurons. When we draw it as one-to-one because that's easier to draw, but in reality, thousands of neurons may influence one neuron, and that one neuron may influence many, many other neurons. And so when one neuron influences another, we call it a synaptic connection. And they estimate 100 trillion synaptic connections in the brain alone. That's more than the stars in the galaxy. So there's a lot going on in there. Your average neuron in diameter, the axon, is no more than 0.05 millimeters in diameter. So let's say if you look at the tip of an unsharpened pencil, to look at a pencil, that's 2 millimeters. So neurons are 40 to 200 times smaller than the tip of the pencil. Now, lengthwise, if you were to line up these 80 to 100 billion neurons in a straight line, it would be about 600 miles long. People often compare brains to computers, but brains are way cooler than computers, I think. Computers can't really deal with a power failure. They don't laugh at a joke. They can't adapt as well as a brain can. And brains invented computers, so let's go with that. Computers do what they're programmed to do, but brains try new things. They try new approaches. And one more thing, if you put more information, a lot of information into a computer, it slows down. But you put more information into a brain, it could actually be more efficient. So brains, yay. That's the take-home message. So let's look at some characteristics of neurons. They are long-living cells. They are as old as you are. These are the oldest and longest living cells in the body. You have your neurons pretty much, unless there's some damage, unless there's something that kills a neuron, you have the neurons that you had when you were born. You still have those neurons. They're actually older than you because they started developing when you were a fetus. So your neurons are older than you are. But the thing is, when they die, most of them are not replaced. Now, the good news is you have way, way, way more than you need. And they've been with you since you were a fetus, but most of them don't seem to be replaced. And for most of neuroscience history, while we're studying it, we do believe that neurons are amitotic. So when you put an A in front of a word, it often means it doesn't do that thing. So they don't divide, amitosis. So most neurons lose the ability to divide. But wait, we really thought that the brain never created new neurons, but until, maybe about 20 years ago, we found that it does. The human brain does create some new cells, but most don't live long or most don't integrate into the working brain, but there are some places. So a word that I will use throughout the semester, neurogenesis. Neurogenesis is the creation of new neurons, and we do have this in human brains. The hippocampus, which we'll talk about soon, part important for memory, learning and memory. Hippocampus is the most active region for neurogenesis. And some things decrease it, some things increase it. So learning increases neurogenesis, the growth of new neurons. Actually, physical exercise does as well. And we do have a burst of neurogenesis in the hippocampus in your first early years of life and during puberty, and now for you, young adulthood. Your brain is developing new connections. Your stem cells are dividing. So we do see neurogenesis associated with learning. We see less of it with depression. So we will talk about neurogenesis later in the semester. All right, more things about neurons. They have a high metabolic rate. So they use a lot of oxygen and glucose because they don't store the nutrients. So they are very hungry. They have to gobble up the fresh oxygen and glucose. And nervous tissue is highly cellular. What I mean by this is, think of bone. Remember bone? Bone is 98% matrix and only about 2% of it is cells. Nervous tissue is the opposite. It's highly cellular. Almost all of nervous tissue is cells. So basic things about neurons. Now let's look at the anatomy of a neuron. And as we see so much in physiology, it's form follows function. Cells look as they do because of what they do. And so neurons look like a long wire because what they do is they carry information. So let's just label the neuron first. And you have one in your notes, so go ahead and label it. These are the dendrites. And one way to think of the structure of a neuron is hold out your hand with your fingers splayed out. The palm, that's the soma, that's the cell body. Now wiggle your fingers. The fingers that come off. So here's your palm, here's your fingers, is the dendrites, and then your arm coming out, that's going to be the axon. All right, so the dendrites are the branches that come off of the cell body. Because if I stick this picture in front of you, there's branches here and there's branches here. Make sure you know that the dendrites are attached to the cell body. The axon terminal, those are the other branches at the end of the axon. And the axon, here is this long wire. Now what covers up this long wire is a substance called myelin. It's a fatty substance that helps insulate the axon, and it's going to make it so the signal carries faster and more efficiently. All right, here we are back in the cell body, also called the soma. And here's the nucleus, and the nucleus is going to create the proteins that become some neurotransmitters and enzymes and important things. All right. This space between the myelin is called the node of Ranvier. So node of Ranvier is the axon, but when we call it the space in between the myelin, specifically the space is called the node of Ranvier. Node of Ranvier is a bit of the axon that is not covered by myelin. And the cells that create the myelin are called neuroglia. There's a whole bunch of different types of neuroglia. The two types of neuroglia that make the myelin, we'll get to this later, but one called oligodendrocytes, those make it in the central nervous system, and Schwann cells make myelin in the peripheral nervous system. So the cell body, let's just do basic functions. The cell body, that is where the nucleus is and keeps the cell alive. And so in the cell body, we have the nucleus. So that's going to code for your proteins and make enzymes and neurotransmitters. We have rough endoplasmic reticulum, which in the neuron is called our Nissl bodies. All right, I probably won't talk about that again, don't worry about it, but rough endoplasmic reticulum, remember that is going to be, it has the ribosomes for protein synthesis. So we need to make enzymes, for instance, a protein enzyme, some neurotransmitters, but mostly it's proteins for intracellular use. We have Golgi apparatus that's going to package up and transport those proteins. We have mitochondria for energy. Maybe no centrioles. Now centrioles, remember, you need for mitosis. And so we always thought, no, there's no centrioles, but now they're thinking, ooh, maybe neurons in the hippocampus do have centrioles because they can go through neurogenesis. Right. And then there's some others. There's some other organelles. There's pigments and other things, but I'm not going to spend much time on it. Let's get to the bulk of the neuron. Let's talk about the other components of the neuron. So the dendrites. The dendrites, that is the main receptive surface of the neuron. What this means is it receives signals. Now, other parts of the neuron can also receive some signal, but the main signals are received on the dendrites. And they literally have things called receptors on them. A receptor is a protein with a specific size and shape, and things are going to bind to it like a key in a lock. And what happens is this dendrite is going to get a signal. For instance, a neurotransmitter is going to be released from the last neuron and bind to receptors here on the dendrites. And that might, for instance, open up an ion channel. So let's say it opens up a tiny little sodium channel. And so a little bit of sodium comes in here and here and here and here and here. And all those teeny weeny bits of sodium are going to travel here through the soma to the axon hillock, is the first part of the axon, and that's going to send a signal, a tiny little change in voltage. And we will talk about that later. But for now, what you need to know is the dendrites, main receptive surface, they pick up signals. And they are branches that come off of the cell body, and sometimes we call it a dendritic tree. And as I said, there can be thousands of connections. The dendritic tree can be connected to thousands of other neurons and influence them and be influenced by them. And how complex it is, how many inputs there are, will affect the pathway, affect how complex the pathway is. Now, these dendrites have what we call dendritic spines. So if here's the dendrite, they have these little protein spines on them. And mostly what these do is they increase surface area, so there can be more connections, more space for things to bind. But I will talk about how dendrites and dendritic connections increase and decrease depending on use. We say use it or lose it. And there have been some studies. So if you increase the use of a connection, so certain neurons are always sending a signal to another neuron, you will increase your dendritic spines because it's like, oh, I need to have more receptive surface area because this neuron keeps sending me signals. So they've done some tests, and back in the 50s, I think this was, and they took monkeys and they stimulated the monkey's left palm, and they just rubbed and rubbed and rubbed the monkey's left palm. And then they sacrificed the monkey and dissected its brain and looked at the right parietal lobe where sensations from touch go. And there were more dendritic spines. So there were more connections on areas that get a lot of signals coming. So you use it more, you form more connections. If you don't use it, you can lose those dendritic connections. Another classic experiment, they took newborn kittens, normal newborn kittens. Make this go away. Normal newborn kittens, and they blindfolded them. And then they took the blindfold off after a couple weeks, and those kitties were blind for the rest of their life because at an important time in brain development, they didn't form those connections and then lost the ability to actually do that ever. So those are dendrites. Next are axons. Axon is the long wire, or I say long, sometimes it's only like a millimeter long or less. Some of them can be a yard long, but some of them are nanometer or a millimeter. So the function is to carry the electrical signal down the axon, where you'll then release a chemical from the axon terminal. And what they do is they conduct impulses away from the cell body down to the axon terminal. That's the only direction they can go. And it's an electrochemical transmission. So it's an electrical signal that goes down the axon. It's a change in voltage. All it is sodium rushes in, making it more positively charged for a millisecond. Potassium rushes out. So an electrical signal goes down the axon and causes release of a chemical at the axon terminal. That chemical is called a neurotransmitter. And this electrical signal is called an action potential. And so here's the axon. And the length of the axon, they vary greatly. Most are a few millimeters long, but they can go just can be less than a millimeter long, or they can go as long as a meter. So there's neurons that go from your spinal cord all the way down to your toe. So if you're Manute Bol, that could be a couple meters long. So there can be some really long axons and some tiny little ones. Most are a couple millimeters long. Now, where the axon meets the cell body, that's called the axon hillock. That's where you start an action potential. And there's going to be a lot of sodium channels there, which we'll get to. The end of the axon is called the axon terminal. And there we have a whole bunch of vesicles of neurotransmitter that are going to be released. And each neuron only has one axon, although some have what we call axon collaterals. So it's only one axon, but some of them actually do have a little branch, and it's considered still one axon. And what this axon collateral does, it's pretty cool. It can feed back onto itself to modulate its own firing. So it's one axon, but sometimes they have a branch that feeds back to itself, back to the cell body, to say, okay, stop firing or increase your firing. It can modulate its own firing. And then I also want to point out again, the axon terminal. It's not just one little nubbin here. It's how we draw it with one little nubbin because it's easier, but there's a lot of them, so you can see how this one neuron might influence many, many other neurons. All right. Now, this is going to be a longer video, go about 20 minutes. This one axon is covered in myelin, and what myelin is, it's a fatty substance. It's going to insulate the wire and make it go faster and more efficiently. Now, you don't cover the entire wire. You have to have these naked spots, these nodes of Ranvier, and I'll explain that in just a minute. So myelin is this protein lipid, speeds the signal and insulates the axon and prevents sodium ions basically from leaking out where you don't want them to leak. And MS, multiple sclerosis, is a disorder of the myelin. It's of the oligodendrocytes. And so you're going to have demyelination of neurons. And so the signals do not travel as well. Now the cells that that myelinate, there's two types of cells that myelinate in what we call the peripheral nervous system. That's all the nerves that go to and from your brain and spinal cord. Schwann cells are a particular type of neuroglia cells that myelinate. And so here's an axon, and this is one Schwann cell. One Schwann cell wraps itself around. I'm going to go backwards for a second. So like this would be one Schwann cell that caused that. That blue thing right there is the Schwann cell that causes to wrap one bit of myelin around the axon. All right. So it's many, many, many layers of Schwann cells. And in the PNS, it's Schwann cells that do that. I'll get to this plasma membrane thing in a bit. Oligodendrocytes, those are in the central nervous system. And one oligodendrocyte can actually myelinate many axons. So it has little arms. And so here's one oligodendrocyte with all its arms. And this one arm creates the myelin that wraps around here. Another arm creates this bundle of myelin. Another arm creates another bundle. So each oligodendrocyte can myelinate up to 60 parts of an axon. Now the nodes of Ranvier, that is where there's no myelin, and this is really important. So here this thing is myelinated, but there's these areas right here where there isn't any myelin. Now what an action potential is, it's sodium leaking, rushing into the cell for a millisecond, and it can't rush through myelin. It can't cross into the axon through these layers, 300 layers of this fatty substance. So the only place it can rush into the axon is at the node and the node and the node. So the way the action potential moves down the axon, it's not like water pouring through a hose. You regenerate a new action potential here and here and here and here and here and here and here. It hops. And the movement is called saltatory conduction. Saltare means to jump. So the action potential kind of jumps from node to node to node. And where the nodes are, there's a ton of voltage-gated sodium channels. There's almost none underneath the myelin because there's no point. Can't use them. And this will make more sense when we talk about action potentials. But a lot of voltage-gated channels at the nodes of Ranvier. And finally, to close this one out, comparing axons and dendrites. Axons take information away from the cell body, and dendrites carry the information towards the cell body. Axons have a smooth surface. They are covered with myelin, but it's smooth. The dendrites are bumpy. They have those dendritic lumpy spines. You have one axon. There are different types of neurons that we'll get to later in the semester, but most of the neurons we're talking about has one axon, even if that axon might have a branch. Dendrites, you have many, 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 many branches. And we say there's no ribosomes in axons, but we're starting to rethink that, so don't worry about that. But there are ribosomes in dendrites. Axons are myelinated, dendrites are not. And axons travel far away from the body, but the dendrites are the ones that branch right there near the body. All right, I'm going to close this one out, and we will continue talking about neurons. We'll talk more about the synapse and the cytoskeletal elements and the plasma membrane in the next one.